Confesc. Christopher Armstrong is a researcher at the Open Medicine Foundation on the use of metabolomics to look at biochemical alteration in MECF patients. He began his work in this field at the University of Melbourne, initiating a PhD project to apply metabolomics to the study of myalgic encephalomyelitis and published his first metabolomics study on MECFS in blood and urine in 2015. Since then, Chris has established collaborative efforts to apply metabolomics to immunological experiments on MECFS, looking at how metabolomics may relate to immune cell function. He has also focused on longitudinal research in MECFS.
Dr. Armstrong in Spain, and good afternoon at your country, far away from us. We want to thank you, deeply thank you for being here today. And the association is really glad that you agreed to be interviewed and to respond to their questions regarding, of course, research in MECFS. So the first question and the second one, which are related, it would be, could you provide an overview? They want to have an overview of the most relevant projects being supported by the Open Medicine Foundations and the expected outcomes. They want to know what can these projects change for themselves. And at the same time, they also ask how these collaboration centers at the different collaboration centers at the Open Medicine Foundations, how are they, how were they selected, or how are they being selected if the selection is still an active process, meaning if these are related to the main aims of the OMF, or is this by invitation? How is this chosen important for the patients all over the world? Yeah.
Okay, great. Thank you. So I can kind of give an overview. I'll start with the first one and then I can transition to the next. So some of the most exciting, I think, research that's happening at OMF globally through all 5 of the research centres. I would start with the first project, which is this long COVID transition to MECFS. We're really interested in this because it's an opportunity to effectively deeply phenotype the trigger at that point in time when it's occurring, at the infection point. And so what we're doing, we're able to collect samples from COVID patients while in hospital during an active infection, and then we'll be following them. So we're looking at hundreds of those, and we're following them long term over a couple of years, looking every few months to take more samples. At the moment, we've taken a few samples. We've taken the samples from the hospitalized cases. We've done a few measurements on that sort of stuff at the moment, on that blood samples that was taken. And then we're also going to be looking at that data as it comes through, and we'll be following those patients to see which ones get MECFS, which ones don't get MECFS, and trying to determine if there are differences between them early on at different stages, when the differences become significant and variant, if there is something early on that can be predictive. And so this is it's a unique opportunity, I think, in the world because of the way that COVID has been, a lot of information regarding this virus has been coming through, and there's been specific things set up to be able to capture at least the active infection during, which is not usually common that you get a lot of people with the flu or other infections where there's this much kind of emphasis on collecting samples and patients with that. So that extra attention has enabled us to do this study, and it's a really good opportunity to try and find out if there is something early on within the infection or the trigger period of the trauma or whatever that can lead to MECFS. So that's really exciting, I think, and that's a collaborative project across all the centres currently through Open Medicine Foundation. Another interesting project, I think, has been, well, just generally, I think some of the interesting projects looking for energy metabolism differences. So a number of the research centres have been looking into this in a number of different ways. You have the nanoneedle, you have a number of different projects through Stanford that are looking for energy metabolism or cell dysfunction issues in ME/CFS. Our group in Melbourne is looking for this very thing as well, metabolic issues, energy metabolism issues, specifically around nitrogen metabolism, or particularly around nitrogen metabolism, I should say. And then we're also looking through, I think in Montreal, they're looking for, or looking at molecular signatures in ME/CFS, looking at metabolism components, looking at microRNA as it pertains to, which are messengers of cell health effectively. And so all these things are being computated. A lot of it is to do with, I guess, cells' ability to deal with stress, but also their ability to create energy as well. I think those pathways are very exciting for ME/CFS. You can understand how it pertains or relates to the symptoms quite obviously, but it's also showing some really fruitful results. So I think those two in particular I'm most excited about. Generally, it was two broad concepts. I think there's also some interesting work through Harvard looking into the neuroinflammation properties in coordination with the groups at Uppsala as well, who are also looking at neuroinflammation and in combination with autoimmune mechanisms in ME/CFS. So, I mean, there are many projects happening that Open Medicine Foundation is funding. So it's hard to give an overview of all of them, but I think those particular subjects are particularly relevant and interesting to me at this point anyway. And in terms of how the centers or the research centers are selected, I think that process ends up happening through the Scientific Advisory Board in discussion with OMF themselves. But I think a clear components of the centres and how they operate is to do with how much support they can garner for that centre, and I think that's an important aspect because it's the way they're set up for Open Medicine Foundation is with the centres. It's about trying to make sure that you have research hubs that work within a community with other researchers collaboratively around them. But those hubs are long-lasting and supported long-term, so that any research findings working with collaborative with those hubs are kind of kept within those groups. And we had noticed a long time ago, and it's a common thing with rare disease, or this isn't a rare disease, but it's funded like a rare disease. It's common where you have researchers who come into that into research that condition. And if they don't find something, they may never get funding to continue on with the project, and they may leave, but that information that they had may disappear. And so the value of what they do with rare diseases and what we're trying to do with ME/CFS, because it's funded like one at this point in time, is to try and capture that information by supporting at least a few small core groups who can work collaboratively with a large set of different people. And so I know myself in Melbourne, I'm collaborating with about, I think I would say about 15 different groups, 10 to 15 different groups at this point. And so that works with me working on them with metabolism, stats, but also providing patients or some funding for the research projects to kind of continue. And so that's kind of how it's set up. But how it kind of initiated in the selection process, I think it's a lot to do with kind of relationships with the other CRCs and research that's kind of in line with what the rest of the groups are finding. And there's a number of different things there as well. But I'm not an expert on the selection process, unfortunately.
Okay, and thank you. So you talk about coordination between the different collaborative centers, and this is happening, I guess, because you already mentioned too that you have data sharing strategies established between the different collaborative centers. How do they are they also coordinated with other centers out of the Open Medicine Foundation, like the NIH or other research centers?
So that's a good question. So yeah, well we have the data sharing center, or the data sharing platform. And that's open to all researchers, not just those on Open Medicine, but being funded by Open Medicine Foundation or part of that collective. That's open to anyone who's researching ME/CFS. There will be data for people to download, and there is already, but there's also the ability for them to upload data onto that if they want to and hold it there. At the moment, that's just one way that we connect with the other groups. We also have monthly meetings to discuss the various projects, and we have also collaboration more common meetings fortnightly with certain projects that we're collaborating on with different groups individually, so within the collaborative research centers. So that's just collaboration within the centers themselves, but also, yeah, there is coordination with other groups outside of those collaborative research centers as well. So there's also all types of research groups in ME/CFS. It's kind of a growing field, and I think it's I'm really happy with how more and more researchers are being involved with this. And so, yeah, we're open with collaboration with other groups. And as I say, I probably outside of Open Medicine Foundation, I collaborate with about 10 different research groups on different projects. And that's about trying to get different people with different levels of expertise to coordinate and work with myself on this project. I'm really interested in kind of using my knowledge with ME/CFS and metabolism. And some people will be interested in other researchers from other groups will be interested, and they may not know ME/CFS very well. They may have a basic knowledge of it. They may have no real access to samples, but they have a really excellent technique, or they're very expert in a certain technique that could be very interesting for ME/CFS to use. And so leveraging my knowledge and contacts to different samples and all that sort of stuff with their knowledge, you can kind of get some really interesting projects. And that's just an example of how kind of collaboration grows from this group. And so, yeah, we are definitely in coordination with a number of different groups worldwide and open for more.
Well, this is a particular issue the associations in Spain centered through CONFES are worried about, that coordination is well established because they want to get the fastest answers to their problems, of course. In fact, the meetings that they are organizing, this is the second occasion for that. In October, they will celebrate this meeting for all the Spanish researchers to present their data, and they are trying to put together a national network. That's why extending this outside of Spain is what they had some questions to know, is this working internationally, or is still being established? So that's wonderful that you clear up this point for them. In addition to ME/CFS, some of the patients ask whether the OMF also has a focus on other diseases. You just brought up that you're doing research on long COVID patients, right? But what about fibromyalgia or other related diseases with ME/CFS? Are these funded? Are projects for these diseases also funded by OMF?
Yeah, so we are, the main focus is ME/CFS research at the moment, though we are looking at COVID as it pertains to how it translates to ME/CFS for that value. But there is, of course, obviously value for post-viral fatigue syndrome, because some of the patients may have long-term COVID, but they may also end up having a post-viral fatigue syndrome-like disease, and not necessarily will they become ME/CFS patients. And also, yeah, we are interested, I think Open Medicine Foundation broadly is interested in a number of different diseases, post-treatment Lyme disease and also fibromyalgia, because they are so similar to ME/CFS. I myself are working on a project with post-treatment Lyme disease as well. though that is not 100% supported. That isn't supported by Open Medicine Foundation, but that's something that is of very great interest to them. They're also working on, which is another actually very interesting project that I think is worth mentioning, which I forgot to detail, which is a development of this personalized automated symptom summaries. That's a project through Harvard, and that's effectively an assisted diagnostic tool for looking at effectively the symptoms of ME/CFS. So breaking down a program that can effectively get detailed information about certain symptoms from patients through leading questions that go on and say, well, you know, they'll, like a GP, they will go through leading questions. So that if you answer yes or no, it will depend on what you answer before it asks the next question. And it uses a lot of algorithms to determine the right questions to ask the patients. But it gets down to very specifically detail out a type of symptom that they have. And the reason why that's important is because that's going to be something that's useful for a number of different diseases that are very similar. This is about supporting patients, but also coming up with a scale effectively that can be used repetitively for repeated measured studies. And that's also really important for me, because I think repeated measured studies, which is what we really do at Melbourne, that's the sort of not the only thing that we do, but this is the key area that we're very interested in, is kind of getting information from patients at individual points in time, getting samples and trying to heavily detail one patient over time so that you can get an understanding of the unique biological components of themselves, while also over time, if you do enough of them, you end up getting subtypes and you develop ways to it. You can tell what parts are unique to the individuals and what parts may be unique to the disease. And so you're looking for also patterns that kind of cross across all the patients. So that's kind of it's called the PASS-CAT program. That's a research project that's happening at Harvard, and that effectively, that test is also going to be looking with post-treatment Lyme disease patients and also fibromyalgia. So that's something that is supported by Open Medicine Foundation at the moment for those other diseases. That's very interesting, yes. So you already said that there is your group is trying to study the possible path through COVID to ME/CFS. But what is your opinion on the possibility that long COVID research helps untangle ME/CFS underlying biology? Well, I think I'm pretty sure there will be some knowledge that comes from the long COVID research that is translatable to ME/CFS. Are you talking about all long COVID projects, or specifically the one that we're kind of working on at the moment? No, I think it's asked in general because broad research. Yeah. I think there will definitely be some information from long COVID research that is translatable to ME/CFS. There are some differences. It all depends on the way that they diagnose long COVID patients, but there will be some that have ongoing damage from the COVID infection to their lungs and heart, and those sort of aspects. They may not be particularly relevant to ME/CFS, probably not. However, there are patients, and we know of many that have ended up having that damage is kind of gone, and that indicators of damage are gone. Their symptoms are more like ME/CFS, so you're talking about post-exertional malaise, fatigue, some of the neurological symptoms that come with ME/CFS. And so those things start popping up. Those are the patients that we're very, very interested in. And those are the ones that we think are obviously, COVID being a significant trigger, ended up initiating ME/CFS. And so, you know, there are a lot of different triggers that can create ME/CFS. And a lot of the thing that they have in common is the severity, the period for which that infection has created a problem for that patient, maybe, you know, over maybe several weeks to a month, which is a lot longer than a normal infection. And those triggering aspects of that initial trigger or trauma seems to precipitate this disease. Not saying that it's the prime cause. It could very well be just the trigger, as we call it, but it's definitely something that will be very interesting, and I'm sure some information coming from that will help MECFS.
Great. So with respect to the metabolic changes that have been observed by your group and also by other research groups, and if we place these metabolic changes in a structure composed by triggers, effectors, or a process of adaptation of the patients, how to explain, of course, to was explaining MECFS pathophysiology, where would you play? Where would you place the metabolic changes along these three players, the triggers, the effectors, and the process of adaptation?
I would say, that's so when effector, do you mean like a chronic stressor? What do you mean by an effector?
So, for example, in a post-viral syndrome, the trigger would be the infectious agent, right? And then the infectious agent brings some effectors into play, which can be one or several, of course, usually several for viral infection. But and then, I mean, it's a question.
And then an adaptation.
Mm-hmm. Yeah.
And you're talking about a host response. So yeah, I particularly take more of the case. I lean towards, or at least for the metabolism, it's the host response in combination with the effectors. So typically, metabolism is an interface between genetics and the environment. So that's where you're looking at most disease. And metabolomics really underlies the phenotype of an individual. So the phenotype is how they express their symptoms. And so metabolomics is the omics cascade that is mostly closely associated with the phenotype. But what makes it difficult to untangle is, which one is causing what? Is it the environment? Is it genetics? Is it the host? Well, it's really just a combination of both. And so the pattern is perturbed from healthy. And so you utilize metabolomics typically to characterize, you can use it to characterize disease, but more typically it's used to characterize a whole number of things like pathologies, where you have a certain type of damage or physiological changes within the patients, like tumors or lesions or stuff like that. And you're trying to characterize based off that biology. But with MECFS, obviously, there's it's a chronic disease, and there are a lot of complications that come along with that. I think the best or the simplest way, we actually call it we call them predisposing factors, which is like genetic factors. We call it triggering, which is like a triggering, like a infection. And then we have maintaining factors, which are other factors that kind of go along with the disease over time. And so probably metabolomics is within that maintaining factor realm. And I guess the way that I think, or we like to think about MECFS, or the way we think it's the best way to describe it is that there is a common biology to explain the common symptoms of MECFS. Most likely, we think a host response, though we haven't completely discluded the fact that there could be a chronic stressor, like a chronic infection somewhere within the body hiding. But we think that the common biology explains the common symptoms of MECFS, but there may be many pathways within individuals that come to that common biology, that create that common biology. And a few of those things include like your genetics, the trigger types that you get, epigenetic changes, immunity issues. Those are the things that are part of those many pathways that lead to that common biology. And we believe that the common biology, or what I believe is the common biology, is the altered energy metabolism in MECFS. And so that's the common pathway, the underlying fatigue. That's from our perspective. But yeah, there could be many paths, many different explanations for this disease.
Yeah, well, I think it mainly was to find out what would be the best way to approach a treatment. Is that if it is directed to something that is not, as you said, bringing up the disease, and you act down the pathway, maybe it's not going to be so effective. So I think that was the question. Yeah.
Okay.
So,
I can, I mean, obviously not a medical doctor, but I think, the current course for treatments are to treat a lot of the symptoms. I think that's the case in Australia and a lot of places around the world, where the GPs will often treat symptoms of ME/CFS, try and help with those aspects. And if that works, and it's something that they keep going with, if it doesn't work, they try something else. It's kind of as simple as that as far as treatments. But yeah, in terms of how effective they are for ME/CFS, they may help with some of the most debilitating aspects and help people live some aspect of their life, or they may not be useful at all. I think it's far more logical to kind of push a trigger towards the common biology aspect, which we would probably call the maintaining mechanism, the host response component for this disease. And that's where we're really targeting. That's where we're really interested. But anything, those sort of treatments for those sort of aspects, I think that takes a long time to get to, unless we can luck out on understanding the biology and find a combination of existing drugs that can get to that. But anything that can really help ME/CFS patients broadly is what we're really interested in as well. So translational, if there are treatments out there that have been effective, Open Medicine Foundation and obviously myself are very interested in how we can set up a clinical trial to test those appropriately and kind of use that clinical trial or pilot trial to help initiate information for countries all around the world to use.
That's great. Well, that should be very encouraging for patients. I think knowing all what is happening. So to finalize the interview, they wanted to have your opinion. The patients wanted to have your opinion on the recent finding by David Anderson's group on the passive transfer of fibromyalgia through the immunoglobulins to the mice. So.
Yeah, I read that a little bit of that work, and I haven't really formed a strong opinion about that, whether it's the case or not. Usually I like to see things like that to be replicated, but I think it's very fascinating that that type of biology could occur. Using mouse models or different types of models comes with a lot of different questions as well when you find that type of research. But I think it is interesting a way, and the fact that people are utilizing different types of animal models perhaps for ME/CFS or fibromyalgia in this case. I think that that's is a promising area to kind of go into. But at this stage, I would probably need to see more information on or replicated findings on that. But it looks very interesting.
Yeah, and they wanted to know whether you think this approach would work to model ME/CFS as well.
Yeah, I mean, it helps to have disease models for research, definitely. It definitely does, because that's the best way for you to trial different treatments and develop different drugs. It's very difficult to play around with humans, obviously. Ethically, there's no way you could really do that. And so that's why they use these models, but also you have the ability to kind of collect different types of tissues from those models, especially if it's an animal model, that may be relevant to ME/CFS to help and find different information. It's just, it's a difficult process. How do you tell if they have the same exact disease and how relevant that is? It's hard to know for me. I'd definitely be interested in ways in which they could maybe work with cells and develop model within, in vivo cells that are collected from ME/CFS patients or things like that. But at this stage, it is difficult, but I think it's an interesting area, and I definitely am not negative about it at all. It's just an area that's probably not within my expertise, and an area that I could recommend or not.
Mm-hmm. Okay, well, thank you very much for clarifying all these questions that they raised. And also thank you from their side. They told me to thank you for the work you're doing and your group is doing and the Open Medicine Foundation is doing. So thank you very much. Thank you very much for having me here, and I appreciate that very much. And I'm looking forward to seeing some new research coming out of Spain for MECFS. That would be fantastic.